Hi, my name is Gabriela Santos Rodriguez. I'm a PhD student at the Garvan Institute of Medical Research, and today's talk is titled Evolution of Circular RNAs in Primates. So one of the main questions in biology has been, how has the complexity of biological system expanded while the number of protein coding genes has remained stable? During the development of genome sequencing, it was expected to find a striking difference between the number of protein coding genes across different organisms. Nonetheless, we can see that across different species with different levels of complexity, where we define complexity as number of cell types, we don't see a dramatic change of number of genes that could explain their complexity. Then, it was thought that the pattern of expression of these genes could explain this complexity. Nowadays, we know, as he reported in Merkin et al., that gene expression of the same organ is conserved across different species, meaning that if we know a gene that is highly expressed in liver in human, its ortholog gene will also be expressed in the liver of other species. As we can see in this heat map from Merkin et al., we have diverse tissue samples from different species from a divergence up to 300 million years ago. And all the samples cluster together according to tissue, as we can easily see in the color code of tissues and figures next to the heat map. Later on, to continue to address this question about complexity, we start analyzing post and co-transcriptional regulation and show us the effect of alternative splicing and intron-retention, where we know that alternative splicing regulates around 95% of multi-exon genes, and intron-retention affects up to 75% of multi-exon genes in mammals. So, given an unspliced transcript, when the process of alternative splicing and intron-retention occurs, we obtain multiple mature transcripts with different isoforms, in which different exons or introns can be included and kept not only in the mature transcript, but also in the protein, which then would have a phenotypic effect. Another product of splicing are circular RNAs, although they are created by a non-canonical process known as back splicing. Here we see the cartoon of a pre-mRNA going through back splicing, where the color-coded bars are exons and the black lines are introns. In this cartoon, we can see that the back splice processing is when a downstream splice donor is joined to an upstream splice acceptor. Such circular RNAs can consist of one or more exons and can even contain unspliced intronic sequences. It is known that circular RNAs are expressed in thousands of human genes, even where the circular isoform expression is higher than the linear one. They are also various expressed in brain and in various organisms. Therefore, it's showing that an increased complexity along different cell types and even across distant lineages. So one of the mechanisms that could aid the expression of circular RNAs are the presence of inverted repeats along the introns forming the circular isoform. So in this cartoon, we again have a pre-mRNA where we have the boxes as exons and the lines as introns. And in this case, we can see that intron 1 and intron 3 are joining due to the presence of allele regions that then could promote the expression of the circular isoform. So also, when studying these molecules across distant lineages, it has been found that there's an expansion of the expression of them across different organisms, where the most striking one is human, where in the case of this study, they found that there would be around 100,000 of circular RNAs expressed. They also found that when they compare the number of circular RNAs or the molecules of circular RNAs between human and mouse, only a small portion of these were shared between them. They found, interestingly, that along the introns forming these circular RNAs, there were repeated elements such as alu and sign elements. So what we are finding during circular RNA expression and its evolution is that there are species-specific events, as we can see, like there's not only like an increase in the expression of these molecules, but also that we are not able to find conserved circular RNAs. So one of the problems when analyzing these molecules across distant species is that we cannot find the impact of circular RNA evolution in cell type complexity. That is also because we already know that cell type complexity evolution has increased in a very short evolutionary time. So then to find what is the impact of circular RNAs in this phenomenon, we need to study them in closer related species. So for that, we decided to analyze a data set of primates species for the divergence times between human and, for example, lemur. It's around only 70 million years ago, and we have tissue samples from different brain regions and different organs. This dataset comes from the non-human primate database and also the CircAtlas database. So as a first analysis, we decided to look into the gene expression patterns, and as previously reported, here I'm showing you a heat map of correlation of gene expression of our primate samples, and we can see that tissue samples are clustering according to tissue expression, as previously reported, where the correlation, the color code is in red, we find them highly correlated, and in blue is low correlation. This result gave us confidence to continue using this dataset as it's able to recapitulate previous results. Then to analyze circular RNA expression, we again took our primate dataset and we used the WIPET program to find in ortholog genes between these species, circular RNAs expressed according to their backsplash junction, where we defined that a circular RNA would be expressed if it had a backsplash junction with at least a percentage of splicing inclusion of 5%, as well as 5 reads. So later on, we decided to see how was the clustering of the expression of these circular RNAs. Here I'm showing you again a heat map of correlation, where again the color code is red, highly correlated, blue is low correlation, and we can see that our circular RNAs are actually being clustered according to species. Interestingly, although these circular RNAs come from ortholog genes, we can see, as shown in this bar plot, that most of the circular RNAs come from a single species. Nonetheless, there's still a subset of circular RNAs that are shared between these species, where we define that a circular RNA is shared between species if the exons forming the backsplash junction in, for example, here human, are also not ortholog exons in the other primate species and are also forming a backsplash junction. So then, according to this definition of shared circular RNAs, we took our set of circular RNAs. Here I'm showing you an offset plot that shows the intersections of our sets, defined sets. In our case is the circular RNAs within each primate, and the lines intersecting these sets are showing the intersection of those sets, and the bar plot is showing the number of elements within those intersections. So then we decided to define conserved circular RNAs if the circular RNAs are present at least in human, chimpanzee, and baboon, as exemplified in the purple arrows in the offset plot. According to this definition, we got around 773 conserved circular RNAs. We also found that within this subset of circular RNAs, most of them were formed from around 2 to 4 exons, where we defined that species-specific circular RNAs are the ones only present in human. So this result also gave us confidence to continue using our dataset because, as previously reported, most circular RNAs are around this number of exons. So later on, we wanted to see the expression pattern of our conserved set of circular RNAs. Here, I'm showing you a heat map of correlation where we have our tissue samples and the primates used for this analysis. And interestingly, we found that the conserved circular RNAs have a conserved neuronal expression between hominoids and all-were monkeys, where hominoid species would comprehend human and chimpanzee, and all-were monkeys would be the macaque species. So one of the questions we want to address was that, what's the impact of conservation versus expression? As previously reported, previous reports have focused mainly on highly expressed circular RNAs, but this high expression of circular RNAs could be related to the high expression of the gene that they come from. So one of the questions that we want to address was if we should continue to focus only on the most highly expressed circular RNAs without considering conservation. So then when looking at the distribution of expression of the circular RNAs between these two data sets, conserved and species-specific circular RNAs, we actually found that there's no statistical difference between their expression values. Nonetheless, when we look at the inclusion levels or circularization rate, meaning that there's a higher expression of the circular isoform versus the linear one, we can see that there's a higher circularization rate in the conserved circular RNAs in comparison to the species-specific ones. More interestingly, we found that there's a relationship between how much circularization rate is as how much all these circular RNA, meaning that, for example, if we look at these circular RNAs present from hominids, old world monkeys, and new world monkeys, we can see that there's a higher psi value in comparison to circular RNAs only present in humans. So we can conclude this, that if we focus in highly expressed circular RNAs without considering conservation, we could bias in getting a noisy analysis, as expression is not the most important characteristic when looking at circular RNAs. Later on, we want to know what features could be associated to these conserved circular RNAs. Therefore, we analyze around 150 features of trans and cis regulatory factors and all major groups of transposons. From we get 3 datasets, our set of conserved circular RNAs, and 2 background datasets. For one was the species-specific circular RNA, and another one was a background dataset formed from randomly combined alternative exons from genes containing conserved circular RNAs. With these sets, we use a logistic regression model to determine the relative contribution of these features. So when looking at the comparison between conserved and background sets, we can see that there's, in line with previous work, the flank introns were enriched with repeated elements such as ALU and L elements in the case of conserved circular RNAs. When looking at the contrast of conserved circular RNAs and species-specific one, we again see that they are enriched with these L elements and also with the lengthening of the intron. When quantifying this enrichment of L elements, we actually see that the L1 element and L2 element is actually enriched in the conserved circular RNAs in conserved to the species-specific one. So also to consider the importance of the lengthening of the intron, we want to know why does this intron had got longer in the downstream exon, and is it enriched in conserved circular RNAs? So for that, because previous work have reported that both introns are longer, we compared the length of both introns flanking the backsplash junction in both conserved and a species-specific one, and we found that the intron downstream the final exon is longer in the conserved circular RNA set. When quantifying this, we actually found that there's a statistically difference of the length distributions of the downstream intron of the final exons in the set of conserved circular RNAs. So we want to ask, why does the intron length of the downstream exon is enriched in conserved circular RNAs? So to contrast and have an answer to this, we decided to take the intron next to this backsplash junction and compare it to the intron, orthologous intron of the lemur that doesn't have that conserved circular RNA. So then when looking at the distribution of our sets of circular RNAs, background, species specific and conserved, we actually see that there's a lengthening of the conserved, of the intron of the conserved circular RNAs, as this cumulative plot shows. Then we wanted to ask, as we have previously showed that we also found that there was enrichment of transposons insertions, we want to know if those transposons has been the reason why the intron has got longer. So then when looking at the insertion of transposons in our sets of circular RNAs, we also found that the introns in the conserved circular RNAs are enriched in transposon insertions. Specifically, we can see that there's an under enrichment in L1 transposons. So with this, our model is showing that during evolution, certain introns can acquire transposons that then therefore would allow the time before the next exons to be transcribed to increase a chance for the backsplicing to occur. So with that, the summary of this talk would be that by looking into the conservation of circular RNAs of closely related species, we were able to account for the rapid evolution of this type of non-coding RNAs. We identify a core of around 700 circular RNAs conserved across million years of evolution, where there's a relationship between its evolutionary rate and a higher inclusion rate. And also that we found that they had a conserved expression in neuronal tissues, at least in hominoids and older monkeys. We also found that conserved circular RNAs have longer downstream introns due to the insertion of novel transposons, and that noisy production of circular RNAs is driven by the insertion of novel transposon in adjacent downstream introns that can over time stabilize to produce conserved circular RNAs. So with that, I would like to thank mainly my lab, EMBL Australia, UNSW, the Australian Research Council, the Garvan Institute, and of course you for letting me give a talk today. So this analysis is already in the bioRxiv in case you want to look at it, and I'm happy to take any questions.